And thank you, everyone. Thank you, Kathleen, for inviting me, and Jenny and Alessandro for inviting me to this meeting. And thanks to all the folks, all of you, but also the folks from CPC that are joining today. One disclosure: the title that I'm showing here is a bit different from the title sent. It was a bit of an overstatement. It said that this was done across the tree of life. I work mostly across vertebrates. Although, yeah, I'm starting collaborations to see what happens with trees and if trees behave a little bit demographically, a little bit as us and other mammals and other animals. To work, yeah. So, well, Colin already said more or less what we do. So in the group, we focus on two ends of research. One is developing methods for these kinds of things. For instance, we develop a method called BASTA, which is an R package for Bayesian survival trajectory analysis that is particularly useful when you deal with natural populations of animals, because there's often problems of not knowing the ages of individuals. And the point of this is to try to reconstruct the age structure in survival and mortality. Being very, very original, we are now developing another package, and the originality becomes with the name. So the other package was BASTA. This package is BAFTA. I hope I don't get in trouble with the name. and it's for fertility. So it's the other side of the demographic spectrum that we're trying to cover. Now it's a package for fertility that also things is when we have these issues of not being able to detect, for example, to know the ages of some individuals or detect all the births. How can we actually do this? But also combining methods that, for example, in ecology and evolution, we tend to be very, very not very explorative in the shape of the age-specific fertility curve, while in social sciences there's a lot being proposed. So the idea is also to combine these two, to merge into many disciplines. And the idea then is to use these methods, to answer specific questions. And many of them have to do with demographic aging, with senescence, with stochastic population dynamics, etc. So, and just to show you a little bit of what we do, but before giving you some background, and of course many of you might be familiar with this, but just to put everybody on the same plane. So, as I said, we are very interested on understanding survival and mortality being the basic building block of survival from an age perspective, and fertility, as I said before. So we're focusing on this. We're interested also in migration, but we treat migration more as a nuisance problem, because we want to be able to overcome the problem of apparent survival that is very typical in ecological data sets. But the idea is to focus on these things. And the point of focusing on these things, on these two vital rates, it's because they are essential building blocks of our understanding of evolution, at least at the population level. So our measures of fitness tend to be linked to these two vital rates. Either if we see them from stable population structure, or if we look at the population from stochastic structure. So this was derived by Tulja Bakar. And the idea here is that the population growth rate, the long-term population growth rate, is a combination of the average vital rates, but also their variability and their covariation. So again, and the vital rates is essentially survival and fertility. So very, very important building blocks. Now, and the point here is that there's great diversity in these vital rates across the tree of life, and definitely across vertebrates. And the point, and this is something that in the 1940s, for example, David Lack came up with this idea that in natural environments, we cannot detect these differences because animals simply don't live long enough. Well, now we have overcome that. Now we know senescence, for example, is quite widespread in nature. So we see that there's very different strategies, and I want to get into this notion of strategies. So we look at these strategies from the perspective of life history, and particularly we try to identify what we call life history traits. Essentially, what they tell us is the timing of key events for the tree of life, as Carol Horwitz here states. And this is just reproduced from Alonso and Kinspeter. And it's just to illustrate that many of these life history traits that have to do with reaching juvenile ages, reaching the age of sexual maturity, the allocation to current and later reproduction, are directly related to these vital rates. No? So, which I'm here, of course, highlighting. As you can see, as you can imagine, and as you know, so many of these life history traits are essentially derived or directly related with these vital rates. Now, broad size, age of sexual maturity, reproductive rates, survival rates, etc., etc. Now, the thing here is that these life history traits, they are not expressed in the same way across different species. So we have a different combination. So here I'm giving very distinct examples. So turtles and tortoises, for example, they may have very steep infant mortality, then the mortality after they reach sexual maturity becomes almost negligible. But they increase their reproductive output with age. While mice, for example, and bats and animals are along these groups that have very, very high mortality, very high reproductive output per reproductive event. And then us, for example, or the primates, we have very low mortality, but we have also very low fertility in terms of the number of birds per reproductive event. We try to allocate our resources in a different way. And this is part of one of the key components. So one of the things that people that are looking into life history theory are looking into is so how the different life history traits covary, or how they relate to each other. And as this is reproduced from Gaillard et al. And it's essentially what we find here is this is a principal component of, these are only three of several of the life history traits that I mentioned. But as you can see, the components actually represent different axes of variation in life history. The first component tends to be related to longevity, to the longevity side, and the second component to the reproductive side. So if we look at the first component and we see where different orders fall, so these are all mammals, we can see that, and this is looking, the first component has become what has been labeled as this continuum, this low fast life history continuum. But we can see that different species and different orders fall along a different section of the continuum. No? So primates tend to be more on the slow end, while rodents and tend to be on the fast end of the And so the way these things covary, and this is the basis of the theory of life history theory, it has to do with trade-offs between your allocation to survival and reproduction. No? And all of these life history traits that actually relate to each of these components. So, and the idea is that this is an optimization problem, that the idea is to optimize your fitness, essentially. No? Of course, and as Noah Express told us yesterday, for those that were in the meeting yesterday, there's a lot of variation within species also. No? And it's important to look at that variability. But we can still look at differences between species and see what patterns we can find between species. But looking at variation within species, one of the things that we recently worked on, this is with the research scientists in the group, Johanna Stark, we looked at one of the puzzling irregularities is this longevity of females, this average longevity of women longer than humans, irrespective of the social context and temporal context. Well, this is actually quite widespread across mammals. It's not entirely new. And we did this also with birds, and we did this with many species. This is not the first study that looks at sex differences, but it's the most widespread study at looking at sex differences. And we do find that the regularity that we find among men and women is also found in other species of mammals. And that regularity is inverted, and I'm not showing here the graph, but the graph for birds is actually very blue. So here it's red because of the advantages for females, but in birds it's very blue. And this has to do with, so one of the main theories for this is the deleterious effect of the heterochromatic chromosome. that predicts actually. The prediction is essentially because in mammals, the heterogametic sexes are males, while in birds is the opposite, it's females. That will explain the general pattern. But there's a lot of variability and there's exceptions that cannot be explained just by this. And we find that they can be explained by sexual selection. No. We are looking also at what happens when there's interventions. So one of the projects that we've been working on is the effect of contraception on longevity, and we find that so the green so the male side is mostly surgical contraception, and for females it's hormonal contraception. These are zoo animals, and we find that there's in general an advantage for contraceptive individuals. And one of the things that we found is when looking at causes of death, for example, was mentioned recently, we find that among contracepted males, there's a reduction in trauma, which is kind to be expected. There's behavior is changed, and to some extent at later ages with chronic diseases. And in females, there's a reduction in infectious diseases. So this is well, it's under review, and we're finishing the revisions for this paper. So this is just some general between species comparisons of what happens, and also within species comparisons. But one of the things that we're very, very interested to look into, and we started doing this in collaboration with many colleagues from the Primate Life History Working Group, is looking at demographic change in primates and looking mostly at survival and mortality. So and the idea is to extend this. So now, one of the things that we were interested to look into, so we want to compare populations of the same species. So looking again at this variability within the species. And one of the things that we set out to do is to say, well, how can we come up with some way of summarizing differences between species and between populations of the same species? And it's by looking at the distribution of ages at death. And if you do any survival analysis, what you end up trying to construct is the distribution of ages at death. No? And just as with any distribution, we can look at extracting summary statistics of the distribution that tell us different aspects of that particular distribution. For instance, as you well know, the normal distribution, if you know the mean and the variance, these are two different summary statistics, and I'm sure that they're sufficient statistics. They can tell you everything, but they tell you two different aspects. One is a measure of central tendency, the other one is a measure of spread. So we did more or less the same. We looked at a measure of central tendency, which is life expectancy, as you well know, is a mean age at death calculated from the distribution. And then we looked at a measure that is related to a measure of spread, which is lifespan equality. And it's in fact, it's the inverse, the log of the inverse of a measure of lifespan inequality. So inequality, it's for example, in economics, it's very widely used to use the Gini coefficient for income inequality. So we use a similar measure, which is known as lifespan inequality, that was developed by Demetrius and then further developed by Kifitz. But we focus on the positive side of looking at equality. So let me explain more or less how these two work. Well, one of them is quite intuitive. So this is just an example on women in Sweden in the mid-1700s and Sweden more recently. And just by looking at these distributions, we can tell they're very different. There's going to be wide differences between the two of them. Of course, the first one, the life expectancy has more than double, which is not surprising. There's very good reasons for that. But then if we try to look to develop an intuition of what lifespan equality tells us, if we split these distributions and see how many deaths happen before a given age, in this case, I chose 60, it's completely arbitrary. But we can see that there's a lot of deaths happening before 60 in the 1700s, in the mid-1700s. But there's also only 6% of deaths that happen in the 2000s. So most of the deaths are now concentrated at older ages. So what our measure of lifespan equality, what it measures is the concentration of deaths at older ages. So, and as you can see, they're quite different. The issue with this measure is that it's unitless, so it's very hard to develop an intuition as we can with life expectancy. But one way in which we can develop that intuition is to just plot a landscape of life expectancy and lifespan equality and see where they fall. So these are the two populations. Again, life expectancy, we know it doubled, quite shocking. how much is too much and how little is too little with lifespan equality. One way of developing further that intuition is to not having only two populations, but as many as we can. So here we have from the very high mortality populations for which we have actually light tables, all the way to more recent populations. And one interesting thing that is happening, and this includes also a time series of Sweden, just as an example, that there is a progression, an increase in tandem between life expectancy and lifespan equality. Of course, what is kind of obvious here is the linearity here. And it can be, the first intuition is this artifactual. It has to do with the way these things are calculated. But if we put more populations of different species in there, the populations, and this was something that puzzled our I'm not a formal demographer, I am an animal demographer, but our formal demographer colleagues that were part of the paper, they were very surprised that these points didn't fall along the line, at the lower ends of the line, were completely possible. So if this linearity, and to some extent we can, by intuition only, we can tell, well, of course, there's been improvements in many aspects of human life, during these periods. So we know that there's been improvements in health, improvements in quality of life overall. But at the time we did this paper, we didn't really try to explain these changes. And also, we couldn't tell if these changes were unique to humans. So this linearity was unique to humans. And then the question is, what drives it? Now, as I said before, we had an intuition, but if it's also not only specific to humans, what could be the basic drivers? And then we can start looking into more mechanistic processes. So one of the things we did is that we did this analysis further, and what we did is we added up many more data sets from wild populations. And then we ended up having several populations of the same species, or at least of the same genus, of closely related species. And what we found is that the linearity seems to be also more widespread across primates. And what's very interesting, and going back to the comment from colleagues, the lines are very unique. They're independent. They don't fall on top of each other. And they seem to be very specific to either the species of the genus. So one of the things that we wanted to understand is, okay, what happens? How can this linearity really be there? And what we focused on trying to tease apart the different components of the mortality profile. So what generates, what could be related to the changes, or what changes the most? And we use a silent mortality model because it actually fits really well mammalian data, and it fits also really well human data, at least the high mortality populations. Of course, in humans, more recently, there starts to be a plateau that is not represented by the silent mortality model, but for the high mortality populations, it does. So again, what we wanted to do is decompose these and see what were the biggest changes. So the interesting thing, this is a competing risk model. So it has a juvenile part, it has an age-independent part. That imagine that you have animals that the risk of death doesn't change with age. Well, that's driven by this parameter that demographers tend to call the age-independent mortality parameter. And then there's the adult part mortality, which is these parameters, these exponential function. Both exponential functions have a baseline parameter and then a rate parameter. And this B1 parameter has often been used as a measure of aging rates, of actuarial aging rates. So in the mortality side. One of the things we did then is to see, well, what happens if, so we are on the line, we take one of the populations, and we look at how much changes in one of these parameters, in each one of these parameters, how much they affect where we are along the landscape if we change the parameter. So this is just an example. So essentially, we're calculating the sensitivity of life expectancy qualitative changes in each one of these parameters. And what we found is that most of the changes along the lines, that you could imagine could keep you along the line, happen for infant mortality and this age-independent mortality parameter. But the senescent part of the mortality, and particularly the aging rate mortality parameter, would kick you completely out of the line. So actually the trajectory of this vector of change is perpendicular to the line. And we couldn't illustrate this with this figure, but the magnitude of change is huge. It's much bigger than many of the other parameters. That's what I just said. So what we try to do then is say, okay, now let's invert the problem and let's see how much each parameter is sensitive to changes in life expectancy and lifespan equality along the line. And this is to actually calculate how much each one of these parameters change. This is relative to the magnitude of the parameter. So this is essentially calculating the area under this curve. So this is a surface sensitivity surface of the parameter changes in life expectancy, lifespan equality, but you need to constrain it to the line. And what we found is that remaining along the line makes you change mostly infant mortality, part of your mortality, but orders of magnitude. So this is in log scale, in log 10 scale. So it's orders of magnitude less the B1 parameter, the changes in the B1 parameter, which measures aging rates. And even for the SHFACs are prosimian, so they might be the furthest from us, from this data set. There's still orders of magnitude less change. At the time, and I changed the name, at the time, we call it the invariant rate of aging hypothesis. Of course, it's not invariant, as you said, Noah, it's not invariant. So aging rates do change and they vary. So we find some level of variance, but it's very, very low variance compared to any other aspect of the mortality. This is just a summary of that, of our findings. And so one of the things we started doing is trying to investigate this in other groups. So we find that, for example, in big cats, we find the same linearity. And I'll illustrate an example with tigers later. But we find the same linearity. These are chronological changes on zoo data. So zoos have improved the way they keep their animals. So if you look before the 60s, they're doing really well. And then as you progress, they start doing better. But they produce the same kind of linear trend as we saw for Sweden, for example. So there's this progression of increasing life expectancy and lifespan equality. But if we go very far away from us and from mammals, and we look at turtles and tortoises. So what I'm showing here now is the mortality of 3 different species, wild and zoos. The red one is the zoo, and on the right side is the aging rates. The right. So the green ones are published data. We didn't have their uncertainty, but we could compare it to the uncertainty that we find in the zoos, and there's huge changes in aging rates that we are not finding in different populations of primates. There's humongous changes. So then, okay, we said, well, we need to delve a bit more into these and trying to tease apart what's happening there. So we're going back to this notion of the slow fast life history continuum, because actually we want to know if different strategies actually tell us something about these changes and help us predict how the changes will be. And just to give you a general idea, so species are right at the fast end of like rats and mice. They, as I said before, they have very high mortality, but they try to reproduce as much as they can. No? So they reach sexual maturity much earlier. And species on the other end are completely the opposite. No? So we tend to take our time, we mature very late, we don't have too many babies, and we don't die that much. Both strategies are, in terms of fitness, they're equal. They give you the same kind of fitness, but they're very, very different. So, and what we did is started looking at some of the hypotheses that have been put forth in terms to understand how these things could vary across the slope of that history continuum. And there's this hypothesis known as demographic buffering that actually comes from environmental colonization. That essentially what it says is that any vital rates for which the population growth rate, which is one of our measures of fitness, is very, very sensitive, you would expect that it will be buffered against temporal variability. So it will vary much less in time. So this is not heterogeneity, this is variation in time across the population. And the predictions are that slow species, like us, like primates in general, there's going to be low temporal variability. So adult survival will be buffered against temporal variability, while in fast species, it will be juvenile survival or even fertility will be buffered. And this is because mathematics of the sensitivity of the population growth rate tell us, oh yeah, since the population growth rate is very sensitive in fast species to changes in fertility and juvenile survival. Now there is mounting evidence for slow species. So this is one of our papers, but there's many other papers that find. So this in mountain gorillas, we found there is a strong signal of demographic buffering in females in particular. So we find that primate females, the survival primate females and young females was very buffered compared to the fertility. Here I call it fecundity, but it's fertility. And the infant side is kind of in the middle. But one of the things is that on the fast end of the continuum, the evidence is not that clear. No, so let's keep that in mind. So one of the things we did recently with Jad Dao, a master student in the group, is to try to be a bit more look more at not just the parameters of the mortality function, but actually the aspects of the mortality. So looking at what is essentially initial infant mortality, the rate of decline in infant mortality with age, baseline adult mortality, and then the rate of aging. It's still using these parameters from the model, but it's combining them in a way that they become even more meaningful in terms of the different aspects of mortality. Now we try to gather published data. One of the things which we wanted to do is to gather published data from multiple populations of the same species. We couldn't find multiple populations of the same species. So one thing that we said is, okay, let's try to group them within taxonomic groups. So let's look at families, what happens within families. And our predictions, based on the demographic buffering hypothesis, is that all of this blue line has to do with the juvenile part of the mortality. We expect that fast species will buffer juvenile mortality, and slow species will not buffer juvenile mortality as much, and the opposite for the adult side of the mortality. And we do find, so these are the juvenile side. So this is on the left is the infant mortality side, and on the right is the rate of change in infant mortality. We do find that the tendency tends to match our predictions. And the tendency for the adult part matches even better our predictions, particularly for aging rates. However, if we look carefully about what happens now, these are lines. We're combining the lines within orders and the overall line, and we find that actually orders are not behaving the same way for the juvenile part of mortality. And one thing that we did is actually we looked also at the coefficient of variation. So we used body mass. Sorry, actually I've said that as our proxy for the position along the slow fast life story continuum. It's been used widely as one of the strongest proxies. And one of the things we did is we looked at, okay, so let's combine what happens, how body mass affects the variability in these components, but let's also not looking only at body mass, but the coefficient of variation within each family. Because maybe what we're doing is picking up the signal of the variability within the family. And what happened is that the two juvenile components, we lost completely the signal. It's not there. The signal remained particularly for aging rates. So it seems that at least from this perspective, juvenile mortality part is not really telling us anything about demographic buffering, or it might not be the process behind this. Now, one of the things with the test of demographic buffering is that they tend to be tested on broad stages and on survival. And this is just an example from Hill et al. This is just two stages, juvenile and adults. Fertility is F and survival is P. Now, if you can look at the temporal variability, and these are your hypotheses. It's either there's demographic buffering or there's no demographic buffering. The problem with this is that on one hand, it's a very neat mathematical exercise because you can calculate the sensitivity from these Leslie matrix and you can get really interesting results. But there's no real insights on what are the mechanisms behind this. And this is one of the things that we want to start delving more into. So, and one of the possible ways of looking into it is, so engineers, they do the survival analysis from engineers is to reliability theory. And one of the aspects of reliability theory is that they look at how the components of a machine It's an oversimplification, of course, how the components of a machine start decaying, and they fail, and how that reflects at the population level. So here's a clock. But and one of the very, very interesting things that happen is that the rate of decline at the population level is mimicking the rate of decline of the components. The level might change, but the rate of decline is very similar, depending on how many components start declining. So what we thought is, let's look at aging rates only. Let's try to focus on aging rates, because essentially what aging rates are telling us, they're giving us a proxy of how these components are declining. And let's try to do that both from the mortality perspective and from the fertility perspective. So our predictions, again, based on the demographic buffering hypothesis, is that for low species, most of the variation will be along the fertility aging rate axis. And for fast species, it will be the other way around. So most of the change most of the variability will be in the mortality aging rates. And that's just a hypothesis. We don't know what happens. We don't know if there's a transition towards that kind of behavior for intermediate species. Just to give you an example, this is again using zoo data, species that fall along different ends of the continuum. So brown rats, very fast species, tigers, kind of in between, in the middle, but tending a bit towards the slow end, and chimpanzees at the low end. And again, these are the temporal trends. We find the same kind of linearity in zoos for all of them. One interesting thing is that brown rats, they tend to change very much along the lifespan equality. And we'll see that it's not only just an effect of scaling in life in terms of life expectancy. If we look at the changes, so what happens if you change aging rates for each one of these guys? So in chimpanzees, the change really, again, kicks you completely out of the line. We already saw that. The paper in tigers is very similar. It actually kicks you out of the line. But in brown rats, and I had to exaggerate a bit this arrow, in brown rats, they keep you along the line. Again, it may look as this is artifactual because we need to rescale everything and make it comparable. If we actually rescale it and we divide all of the life expectancy by the maximum life expectancy, one of the things that we see is that the direction tends to be the same in all cases, but the magnitude is definitely not. What we find is that, again, if you're a chimpanzee and I change a little bit your aging rate, you become a completely different species. The same for tigers. But for rats, it doesn't matter. No, the effect of changing by the same magnitude, the aging rates is absolutely negligible. So most of the changes are happening in all aspects of the mortality. So this actually matches what we expect. And these are the posterior densities of the parameters that actually create this. So we find a lot of variability in brown rats and much less variability in the groups. And this is consistent with what I already showed on aging rates at the family level. Now, fertility, it's been a bit harder because it's interesting because fertility is something that we can measure repeatedly, but unfortunately, there's very few published data on fertility. So these are some of the published data that we found. We use BAFTA on them, and we were able to reconstruct the fertility, but we didn't have enough data to be able to do the same kind of analysis across different species and across family, for example. So what we're trying to do now is to look at for example, data from the primary life history database. We can actually look and in other populations of long-term studies and look at differences within population. But again, the idea is to measure aging rates. The problem with fertility in mammals is that it doesn't behave in the same way as mortality, that you have one model that fits very nicely everything. So in fertility, as you can see from this graph, it varies a lot. So we need to calculate derivatives and log scale, etc. Log derivatives. So this is just a quick overview because these are some of the published data that we could get our hands on. And so these, again, within primates, these are changes in aging rates at specific ages. But as you can see, as you move from baboons to humans, these are the Hadsta hunter-gatherers. One thing that you can see is that in baboons, depending on where you measure it, but aging rates for most of the course of life, fertility aging rates are very low. They're very short, the magnitude of change. And the mortality aging rates are much larger. As you move towards humans, so in chimpanzees, they tend to to be very comparable, but in humans, most of the change is infertility aging rate. So there's something happening there that is quite much more dramatic than we expected. Okay, so in terms of future direction, so these, again, this is a work in progress. We're trying to get more data, but we also want to look more at what are the mechanisms. And as I mentioned before, demographic buffering actually stems from environmental canalization. And when Waddington and later a lot of work from Stearns and colleagues was looking at more of the mechanisms and the evolutionary aspects of how the variability in a given trait could be controlled, could be reduced. So the idea is to answer a couple of open questions that will require a lot of work, and many colleagues are much more knowledgeable than I am. So first of all, how could this have evolved? If we find the trend, if we find a pattern also on the fertility side of aging rates, but even see if actually this could be the result of evolutionary processes, and what could be the actual underlying mechanisms, no? And that's where I'm completely lost, but I have very good colleagues and I hope to get more colleagues. So in terms of evolution, so one of the things, of course, the first thing that we're doing is looking at, well, what happens? What is the sensitivity of fitness measures, population growth rate, for example, to changes in aging rates, particularly. I don't have the results yet, but we're looking into that. That's a mathematical exercise. No? But then we want to couple that with some of the predictions of the evolutionary theories of senescence, no? Mutation accumulation or early life trade-offs as predicted by disposable soma theory or antagonistic pleiotropy. Then this is where I'm completely lost. And I'm relying, and actually I'm very open to discussing this. It's looking at the underlying mechanisms. One of the things that, and this was in response, there was a comment that was going to be produced for the paper in Turtles that I showed you from one of the very big names in the evolution of aging, which was that there's no way we can, there's these changes in regulation that will make you have lower aging rates. And that we will individual. But in fact, there's been some evidence that the expression of some genes in fish, for example, they can be genes that are related to aging rates. They can be downregulated or upregulated, making aging rates very different between captive and wild populations. And there's the notion that there's also low conservation of gene expression at older ages and mutation accumulation. So I would like to start looking at these things, but I know that's daunting because the number of things that can be related with gene expression or which are the genes are, and I'm showing these graphs. These are the hallmarks of aging, just to illustrate that we're very aware that it's a very, very complex process. And then, of course, following the talk yesterday, looking at other possible mechanisms like declines in methylation levels. But then, yeah, these are open questions that we are trying to find collaborators. We have snatched Jenny already to be part of this. And before I finish, I would like to thank my direct collaborators, Jenny, Tung, and Susan Alberts, the department that has given me the opportunity to really have time to think about all these things, the Department of Primate Behavior and Evolution of the Max Planck Institute for Evolutionary Anthropology, my group, which Johanna Stark, Jad Dow, Johnson, and the Primate Life History Working Group, because they are our first initial collaborators on this project. I don't know if I went too fast, but thank you very much.
Go ahead and field questions.
Thanks, Fernando. I feel like I'm learning a lot from this talk.
Oh, I hope that's bad.
We're trying to. I'm curious. So for the idea of demographic buffering, is part of the hypothesis that environmental variability itself is selecting for demographic buffering and like a stabilization in vital rates?
Yes, it's related. Yeah, and that's why they didn't relate it to genetic canalization, but to environmental canalization, because that's the idea is that yes, it is related to environmental change. But the thing is, and let me see if I can answer correctly this. Yeah, there should be a genetic component to it, because one of the things that the theory predicts is that you shouldn't, those vital rates that are very key, they shouldn't vary too much. You know? And that's why we're interested in looking at the mechanisms. Because yeah, demographic buffering itself hasn't really delved that much into that. But it's, yeah, I guess a combination of the two. I think my answer is not very convincing, but yeah.
So I appreciate that. I think the fertility data you showed was female fertility.
Yes.
Yeah. And I appreciate that female fertility is going to determine population growth rates. In terms of aging, though, there's also fertility senescence in males.
Yep.
To some extent, I was wondering if those show really different patterns, or I guess specific human data with menopause versus lack of.
Yes,
I'm just curious if you see similar patterns that just shifted for the male data, or what that looks like across different species.
yeah, so we did that the study with gorillas, we did it also for males, and I didn't show the graphs, but we do find differences in fertility, that strong difference between males and females, and they're very driven by the environmental conditions. So we did an analysis between different populations of chimpanzees, and we find that fertility is very limited. In terms of menopause, it's hard to say, because most of these species, it's very hard to detect menopause or that they reach that point. There's been a paper recently on potential menopause in chimpanzees, but if you look at what they call the post-reproductive representation, is very, very short still. No? So, but there are differences between males and females in terms of fertility, very steep difference.
All these trade-offs you're talking about, the population level, do you think they may also apply to the level of individuals within species?
Yes, I think so. And you know, it's been interesting, because when you try to detect trade-offs in wild populations, it's kind of hard. And because there's the notion of heterogeneity. You know, there's a problem of individual heterogeneity. It's one of the aspects that we're interested in. Is trying to separate based on quality, but in terms of frailty, analyses are called the quality of the individual, which is what's its capacity to survive, what is its capacity to reproduce. But there's differences between individuals, but you would expect that if you look at the same individuals with exactly the same conditions, well, they would be affected in the same way. Respect, yeah.
Instead of age-specific fertility, have you thought of using lifetime reproductive success as something more relatable to fitness?
So that could be a measure of fitness itself. But what we're interested in, so we could use that as a measure of fitness. It combines both measures. It needs to combine fertility and survival. Definitely as a measure of fitness, for sure. Yeah, we're not just married with population growth rate. It can be, yeah. Mm-hmm.
This is kind of specific, but we'll talk about this tomorrow when we talk about our workshop. But as it's becoming pretty evident broadly that primates in captivity in our labs have longer lifespans than the primates in the wild, for a lot of obvious reasons. But it's creating a puzzle for how to compare timelines and aging processes in the different contexts. So I just was wondering what your initial thoughts are on.
Yes. And actually, that is something. So when we did, let me go back to these graphs. But when we did this initial analysis, well, the extension of these analyses. So these actually some of these data points are from captivity. And when we first were discussing, we were inviting PIs that had long-term data sets. We told them, well, maybe we can use also captive data, and they were like, no, that's going to be comparing pears and apples. Anything to do. But they just fall along the line. So they are demographically, they're still the same. Now, in terms of pace of life, yes, so their longevity is extended, but it's extended most likely because they're compressing their mortality at older ages. And it's the same that happens in human populations. If we try to compare human populations across time, it's the same thing. So I think they're still comparable. it's still the same species. They don't become something else. They don't become aliens like my colleagues were thinking.
Right.
Demographically, they're the same.
Are you saying there's maybe a tipping point at which we, like, beyond what would be a wildlife expectancy, we would be looking at a different aging process in a captive animal than a wild animal, or do we consider it through their whole lifespan? As someone who's really unfamiliar with how demography works.
Yeah, let me see if I understand you. But oh, yeah, can you read?
I'm just trying to clarify what you just said. So are you saying that, like, if there's a shorter lifespan in captivity
Mhm.
or in the wild, is that like the age at which we should be thinking of anything beyond that in captivity is a different process of aging that we're
Oh,
looking
okay.
at? That, no, not that.
I see. Yeah, I guess
I know there's no clear answer for this.
no. And so, yes, and the processes that kill animals in the wild are different from the processes that kill animals. So many of the environmental factors that will kill an animal, they're not in the zoos.
Yeah.
And you start seeing, for example, for the contraception paper, we started looking at causes of death.
Mhm.
And you start seeing many of the causes of death that you will see in
Yeah.
other humans. Start popping up cancers and things like that.
Right.
And those will not show up. But one of the things, and this was one of the discussions that demographers and evolutionary demographers tend to have, is how much can we actually extend our lifespan? And one of the things that we're finding here is that in order to really now, if you look at Sweden now, Japan, in order to extend our lifespan, the only thing we could do now is reduce aging rates, and that's something that we cannot do. So, and I know that it's a very coarse measure of many things that are happening under the hood, and that's why we want to look at those things. But yeah, so the comparison between zoo and wild, to look at what are the things that are killing individuals, for example, it's very well known.
Can I just ask for a clarification? I think my understanding, I should have a good understanding of this from the paper, is that when you say you're demographically the same, they're not demographically the same in some of those parameters. They're just demographically the same from the rate at which mortality increases in adulthood. Just the rate of aging parameter, that's what gives rise to that line.
Yeah.
So if you had captive and wild primates, they probably do have different values of baseline mortality, right?
Yeah, they do. They do.
Have different levels of juvenile mortality. They
Yep.
do have different levels of decline in mortality in early life.
Mhm.
But it's that crucial, like, how quickly does mortality go up? That's doesn't seem to change within closely related species.
And you know, so it's kind of a semi-experiment. So in zoos, you have many animals that come from the wild, for example. You know? And if you look at their longevity, yeah, they're not as longevous if they came from the wild as the ones that were born in captivity, but they're more longevous than they were in the wild. So if you take them and put them there, or an individual that is born in the wild, you immediately put it in captivity, it's very likely that its longevity is going to be, its risk of death is going to mimic the one of those that were born in captivity.
Right.
So, and that's what I mean in terms of, if I grab an individual in captivity and put it in wild conditions, it's going to die just as the wild one. And but what's, as Jenny said, what's interesting here is that aging rates don't seem to change much in these groups. In other groups, they do change a lot.
Right.
That's good. Probably a very stupid question. Speaking
no
about
stupid-
the
there's stupid
mechanisms.
answers, but not stupid questions.
Speaking of mechanisms of the rate of aging across species, I recently became aware through a collaborator that as male of our species age, will have a somatic loss of the chromosome Y in certain percentage of cells.
Yeah.
And that appears to be associated with the rate of mortality and diseases. Can that make any sense in the context of your analysis? Because
Yeah,
of
for
course,
sure.
the loss of the chromosome Y would be associated with reproductive.
Yeah.
I shouldn't say it. Reproductive capacity, maybe.
Yeah.
Reproductive rate, I don't know. But appears to be relevant mechanistically for age-associated conditions and mortality.
Yeah, for sure.
Can this make sense in the context of your, you have a large.
Yeah. Yes. It can. And as I said before, I'm just delving into these kinds of things, more of the physiological and genetic mechanisms. But it should, I think, or at least it's one of the things that could be explored. The problem of finding potential mechanisms is that it's so complex that finding a single mechanism might be very hard, but definitely. So we need to look into
But you have
these kinds of things.
tissues or genetic data
Well-
on those individuals?
Well, not yet. Yeah. So one of the things that we would like eventually to do is establish collaborations not only in the wild but also in zoos to get to see if we can get access to those data. But it's I'm just starting to kind of frame these things, and we need to look into mechanisms that we can actually measure both places. But yeah, definitely.
So sort of demographic buffering with aging rates, that is indicating, like Nick was asking, it's suggesting that you can change the environment a lot,
Yep.
right? But you don't change the rate of aging.
Yep.
And so a lot of the conversations we've had here so far and things that people are interested in are changing the environment, studying how change in the environment change things.
Yeah.
Right? Whether they're measures of methylation or gene expression,
Mhm.
oxidative stress, or like whatever.
Yep.
Right? And so if we were trying to understand the mechanisms underlying the fact that the environment does not change,
Mhm.
according to your results, the rate of aging, should we be looking for physiological mechanisms that are themselves very buffered from environmental variation? Like we throw all sorts of things at it and they don't do anything? Or should we be looking for things where you throw a lot of things at it and they actually do a lot because that plasticity is what gives you buffering?
Yeah. Well, first I think we will need to think how much they relate to the rate of aging, so to aging itself. But yeah, so methylation and yeah, definitely. You said that plasticity generates buffering.
No,
No,
it
it's
can,
contrary.
right? So if the temperature goes up
Mm.
and your body does nothing physiologically, then your survival will not be buffered.
Mhm. I see.
Right?
Yeah.
So a lot of things that allow you to
Yeah, for sure.
retain some sort of stability on the high level you're looking at
Yeah.
require a lot of
What are the
dynamic
responses to it?
change
Yeah.
at the physiological or molecular level.
Mhm.
So when I'm thinking about, like, if you're like, what are the mechanisms? I'm trying to figure out what the criteria for good candidate mechanisms would be even be, right?
Yeah.
You change a lot with environment, you change not at all with environment.
Mhm.
And to be honest, I think that a lot of these things, including stuff like DNA methylation, doesn't map to aging rates. It maps to maybe mortality if you're lucky, but that can all be the invariant component and not the rate of aging at all.
I see. Yeah.
I don't know of a lot of interventions. That there was this old Linda Partridge paper
Yeah.
where she said all the things that we do to Drosophila that change how long they live,
Yeah.
none of them affect the rate of aging.
Mhm.
They all affect other
Yeah.
things in your demographic.
Yeah.
And I don't know if there's an update of that since then. Other people here might.
Yeah, from what I know, no. So at some point, changes in temperature were associated with possible changes, but yeah, it's true. It hasn't been found. Yeah, and that's a very good point, actually. Yeah, I haven't really thought about it. So I will take notes and think more about it. Yeah, it makes absolute sense. To look at how plasticity, what are the mechanisms that vary sufficiently so they can remain, the aging rate remains the same. Yeah, sure. Think about. Yeah. You had a question?
Yeah, it was kind of going off of Alessandra's comment on the loss of Y. And I just thought, is that about the same thing looking at one of your earlier slides where you were examining all the mammals for sex differences in lifespan?
Yeah.
And I think you call that
Heterogametic sex.
exactly. So I was just going off it and thinking that could probably be related to the loss of.
Yeah. It could be, yeah. So yeah, and the two hypotheses that we know of have to do with either the toxic X, the toxic Y, or X. So it could be, yeah. We haven't really looked into it, but possibly. Yeah.
I was much interested in a different thing you said about this slide, so that's good. You said that some of this variation was explained, within mammals was explained by differences in sexual selection.
A lot of that.
What does that mean? Like once you measure.
So, and looking mostly at mating system. So, monogamy in males actually reduces the sex differences and increases the sex differences in birds. And it's a well, it's somewhat of a known or proposed mechanism.
Yeah.
So here we find that actually it's more even important than, for example, the cost of reproduction.
Yeah.
Cost of reproduction, it becomes relevant, depending on the measure that you take, becomes relevant in birds, but not as much in mammals. Part of it is that there's very little biparental care in mammals and a lot of biparental care in. But yeah, so mostly it's related to sexual selection through also a measure of investment in sexual characteristics, sexual sex dimorphism.
I'm now seeing the purple and the yellow. I'm just thinking if you could potentially use a more continuous metric, right? Like the difference in reproductive skew between males and females, right? And situations where you had more reproductive skew in females, I'd expect male advantage.
But I think it will be heavily related to mating system. So
I think, yeah, I definitely have.
yeah.
Right,
Yeah.
just,
And ideally we didn't have those measures for all of these species. So we had to really look at very coarse measures.
okay.
Yeah.
Thanks.
But yeah, it makes a lot of sense. Yeah.
I'm still trying to understand a lot of what you explained. Thank you. But when you compare those slow and fast species on the continuum, does it then mean that you expect greater inter-individual variability in whatever mechanism explains like aging at earlier age in a long-lived species? And if we would to measure that same parameter in a fast living species, you would actually expect greater inter-individual variability in adult life? Or yes?
Yes, and so normally you do this comparing populations of the same species or comparing them in time. So you would expect that, yeah, for sure.
And do we have any parameters that has actually been measured, whatever it is almost, that shows?
For individual heterogeneity?
Yeah.
Well, so there's the frailty models that you can use. So the issue here is that you have no measure of individual heterogeneity. And when you try to analyze some in species, but you can test a model that actually has implicit individual heterogeneity, which is the Gamma-Gompertz model. That model, there's one parameter that actually measures variance in the individual heterogeneity under certain assumptions from that. Differences are proportional, so individuals are born like that, they don't improve in time. We have some deviation from that where we can try to look at individual heterogeneity in aging rates as well. We haven't done it, but that's one of the things that we should actually test, individual heterogeneity in aging rates.
But this would still be, you mean, demographic, or you use some kind of physiological measure? Or...
No, it's purely demographic.
Okay, so I mean,
you have to extract it from the data. So
so we don't have like a parameter that would show kind of a matching great heterogeneity in early life within long-lived species that is kind of constrained later on, and the opposite in
the modeling in early life, no. So separating by parts of your life course. Well, we could look at it from the adult side, if there is individual heterogeneity. So what happens is that the mortality curve slopes, so reaches an asymptote. So it's this S function, like logistic function, and it's known as a logistic model. It's a result of the effect of individual heterogeneity because there's selective disappearance of individuals. We did test that model on all of these data, and it didn't really perform well. So, but only on the adult side. So it was the sidle model that performed the best. So we couldn't actually detect a signal of individual heterogeneity, and it's difficult because for some species we had a handful of individuals, so it's hard to pick up the signal.
I think we're out of time, but Fernando will be available for those who have additional questions. So thanks very much.